Hi, my name is Elizabeth Powers. I'm one of the co-leads for the Terrestrial Ecosystems team. Today, the focus of our webinar series is going to be on microbial ecology in permafrost environments. So what I'll do, we have two speakers today, and I'll start off by introducing our first speaker, who is Mark Waldrop. He is a research scientist with the US Geological Survey, and his research focuses on microbial ecology and biogeochemistry. I believe he got his PhD at UC Berkeley in microbial ecology and biogeochemistry. So today he's going to give a presentation on microbial ecology in permafrost environments. And then he'll give about a 20-minute presentation, and then we'll turn it over to Neslian for her presentation. So with that, before I turn it over to Mark, I request that instead of doing a verbal roll call, if in the chat you can introduce yourself by adding your name and your affiliation in the chat. All right, thank you, and take it away, Mark.
All right, thanks, Elizabeth. And my PhD was actually in soil science, so it's rare to see a soil science PhD anymore. They don't exist in too many places, but that's what it is. So I'm going to share my screen and now pop up my presentation. Can everybody see that? Okay.
Yes.
All right. So to begin, obviously the permafrost landscape is vast. It's carbon rich and it's thawing. It covers 23 million square kilometers of the Arctic and contains approximately 1,600 petagrams of carbon. And widespread permafrost thaw is expected to occur over the course of this century, potentially releasing 1 petagram of carbon per year near the end of the century. So this really makes the permafrost environment one of the largest biotic feedbacks to climate warming. And this graphic on the left is from Turetsky in 2019, in red showing areas that can undergo abrupt thaw, which would mean it's going to be thawing even more rapidly, potentially releasing both CO2 and methane and thermal karst bogs. But what I'm going to be talking about today is the fact that understanding the biogeography of permafrost microorganisms is important because the organisms present in permafrost could have some potential impact on biogeochemical processes before, during, and after thaw. So that's a little bit what we're going to talk about. Permafrost, the biogeography of permafrost microbial communities has really not been examined at scale. They're obviously, these communities are sharing many common stressors. There's extremes in temperature, there's lack of diffusion. The microbial environment is sort of shown here in a scanning electron micrograph on the right-hand side, showing these little brine channels that may exist in soils where the microbial cells may survive. Permafrost communities are derived from a wide variety of habitats. But they're often, they can be very deep and they can also be very old. And there could be some evidence. It's possible that microbial communities may be reflective of past environments or past permafrost conditions. And we'll touch a little bit on that today as well. So in this presentation, I'm going to be talking about the environmental factors that are structuring the composition and functioning of microbial communities across the Pan-Arctic. And I'm primarily going to be pulling data and information from these two recent articles that we published on permafrost microbial communities and some of the ideas generated by the Permafrost Microbiome Research Network on how microorganisms may in fact affect processes post-thaw. Our laboratory works on a lot of gaps in knowledge related to the permafrost carbon feedback, where we really focused on rates of microbially mediated biogeochemical processes among various ecosystems, as well as the spatial distribution of microorganisms and low temperature microbial activity, or that is microorganisms active in warm permafrost or just at the freezing point, And that's just a little bit of background about our group. And looking forward, some of the discussion questions that I think we could maybe talk about at the end of this, if there's time, is who wins when permafrost thaws, and do permafrost microbes affect ecosystem behavior post-thaw? Additionally, can the genomic information, which we're going to be talking about a little bit today, be used to tell us not only about microbial functional potential, but can it also tell us about future ecosystem responses and maybe even the paleoecology of particular sites? So microorganisms carry out a wide variety of functions in soils, and many of these functions can be described through the field of eDNA or metagenomics, where we can pull out functional genes from soils, and by looking at the relative abundance of those functional genes, they tell us something about the potential activities that these microorganisms can carry out. Microorganisms degrade organic matter, obviously. They ferment, they conduct fermentation, acetogenesis, methane production, and a wide variety of terminal electron accepting processes such as nitrate reduction, which would result in nitrous oxide or denitrification, iron reduction, and sulfate reduction. Of course, when there's oxygen around, they're going to be taking part in heterotrophic respiration and decomposition. So it's a wide variety of functions, and most all of these functions can be examined through the DNA that we can pull out of permafrost. Moving on to another important concept that was brought up in this Ernakovich paper in 2022, that there's a lot of ecological theory that can be used to describe the spatial variation in microorganisms that we might observe in the environment. And these are things such as sort of the random assortment of microbial communities, stochastic processes. There could be diversification of microorganisms that occur in soils over long periods of time. There could be dispersal limitations that, especially in an area like permafrost, where there's a sort of a lack of dispersion that could affect the composition of microbial communities that we observe on the landscape. But the primary focus that our group takes is really related to these deterministic processes or selection, and that is that there are environmental factors that are going to, such as climate, soil carbon, resource availability, pH, and things like that, are going to select for types of microorganisms in the environment. And therefore, by understanding this environmental metadata, we can better understand what controls microorganisms across the landscape. Now, next slide. All right. It's also our idea that microorganisms in permafrost were originally sort of laid down by the paleo environment, that they were reflective of the climate and the soil properties thousands to tens of thousands of years ago, just before that permafrost was frozen. So there's that factor that affects permafrost microbial communities. But then there's the additional factor, which is fairly unique to these environments, which is this time factor, the paleo time. So as these microorganisms are entrained in permafrost, are there changes that occur in community composition or function as these soils have aged? And so we can try and examine that in part statistically by just understanding what is the date of onset of permafrost formation within these various locations that we sampled around the Panarctic. So we'll talk about that a little bit. Oh, and I think am I blocking part of your screen? So we worked with a wide variety of investigators from this permafrost microbiome network, and we sampled 133 soils around the Panarctic. And it's just important to point out that these soils came from a wide variety of systems. Here are just some pictures on the right showing some of the variation in soils that we might observe, from very low carbon, high Arctic weathered siltstones to frozen fens and bogs with lots of frozen macromolecular organic matter, to yedema, which would be very thick silts with high DOC contents and things like that. So there's a wide variety of ecosystems, and we tried to pull as much metadata together as we could from all of those. And then we were able to, then we could pull DNA out of our soil cores, and using metagenomics, we can then look at the 16S ribosomal RNA gene, which tells us something about the taxonomic composition of these microorganisms, where then we can look at the relative abundance of various phyla as well as their diversity. And then we can also look at the functional traits of microorganisms using a KEGG database, and these are the functional genes that should be coding for particular proteins that carry out processes within soils. And in the end, we can use a cluster analysis to see if microorganisms or their functional attributes cluster together, and then can we tell something about the way that microorganisms may function post-thaw by the way that these functional genes have clustered. So see, these are some of our taxonomic results. So looking at 16S ribosomal RNA, these are all of our sites on the left-hand graph here. The dominance by the dark blue and light blue indicate that there are some commonalities in taxonomic composition across all these sites, indicating that proteobacteria and actinobacteria are typically the dominant members of the microbial community across the entire Panarctic. Perhaps not so surprising. And of course, there's lots of variation in various phyla in this space of very low relative abundance. And there are very few outliers. For example, the Fox Tunnel up here. I'm sorry, not the Fox Tunnel. I forget the location. I forget the name of that site. But some of these sites do look like outliers, whereas others. Anyhow, there's a commonality of the dominant members of the community. Another important aspect of this research showed that there's a unimodal relationship between soil pH and microbial diversity across all sites, where microbial diversity is typically maximized near intermediate or neutral pH. And this is a common finding from lower latitude terrestrial environments, and also observed here within the Panarctic. Now we get into a little bit more about the functional genes. So what we have here is the relative abundance of the functional genes that differed across the most across this entire data set. So it's a reduced data set from the entire functional gene data set. And we wanted to see whether these functional genes then cluster within groups. And so we do observe a clustering of functional properties, and these resulting in 6 main clusters, where we have 2 up here that have genes that are in high relative abundance related to methane production. So these are kind of our 2 methanogenic clusters. We have several clusters in the middle that clusters 1, 2, and 3 that have a large abundance of functional genes related to methane oxidation, nitrate reduction that can produce NO2 or N2O, and cold survival. And then we have this one very large cluster that's primarily from the subarctic. That is the one thing that really distinguishes this from many of the others is the presence of iron reduction, and which is becoming more and more interesting in Arctic permafrost studies right now. So then if we take this functional gene data, and I'm going to show you, we wanted to say, is there a spatial, well, actually, let's think about it. What are the factors that may control, what are the environmental factors that may control the distribution of these various functional clusters within the environment? And so we took these different clusters and we put them on this graph that is basically showing us the average of certain metadata factors that were the most important for controlling variation. So the first thing to note is that factors such as latitude, which we believe is a proxy for climate, soil pH, and soil depth were the 3 primary factors that influenced the variation in these functional genes across the Panarctic. And in this graph, The size of the bubble is not the size of the variation. It's the depth from which, the average depth from which those samples came. Whereas where it falls on the pH graph is the average pH for all those samples within that cluster. And there are differences among these various clusters in their latitude and pH and their depth. But what we observe is that these two circumneutral clusters, clusters 4 and 5, are our two methane producing clusters. These three clusters that are sort of a higher latitude and more acidic pH are the clusters that have genes involved in nitrate reduction, survival of cold conditions, and methane oxidation. And then this one, cluster 6, is the one that was had functional genes that were reflective of iron oxidation. And this is typically from sort of a deeper soils where there might be more mineral content and more iron reduction. So the take-home points from this are that the biogeographic variation, there is biogeographic variation in taxonomic composition and in functional traits of these permafrost microbial communities across the Panarctic. The communities, but in particular their functional attributes, are structured dominantly by three factors: latitude, again a proxy for climate, soil pH, and soil depth. Other factors, primarily permafrost age, but also carbon, nitrogen, did have some small statistical effect on community composition, and so that there is some evidence that there is a link or a signal of paleohistory on microbial communities in these environments as well. And I think this study also shows to me that soil pH is a really important factor structuring the functional potential of microbial communities across the Panarctic, and it's probably something that should be examined more, especially with regard to methane feedbacks and things such as that when permafrost begins to thaw. Now to sort of get into a little bit of thinking about some discussion points and what this might mean for future studies, is that some of the real big questions in this area of research is so what if we understand that there's this distribution of microbial communities and their functional traits? The question is, like, upon thaw, does that actually matter? So is there some impact of that permafrost community that we know something about on future active layer dynamics as that permafrost community begins to thaw? So are those members simply do they simply die out and are consumed, or is there some influence on the presence or absence of microorganisms in permafrost on future processes post-thaw? So this is some ideas that came out in the Cernakovich paper 2022, and so I think these are important areas of future research. And so I just want to end by thanking my work community and all of the people who are influence my daily life, especially emphasizing the Permafrost Microbiology Research Coordination Network, which is, makes a lot of this kind of stuff more and more possible. So thank you very much.
Hi, Mark. Thank you. That was a really interesting talk. We have a second speaker, but what I want to do before we move on to the next speaker, we'll have a dedicated discussion session after the second speaker. But I wanted to open up to see if there's one or two specific questions related to Mark's talk, and then we can open up for discussion after the second speaker. So we'll give a moment if somebody has a question for Mark.
Rachel says those are the oddballs. Oh yeah, Vault Creek Tunnel, yes. Mm-hmm.
Well, Mark, I have a quick question for you. I was curious, you looked at several drivers of latitude, soil pH. I'm wondering if you looked at the role of vegetation type or plant community type, if that influences the microbial community at all.
We haven't done a good job at that. I mean, most of the factors we were looking at were were sort of continuous variables that we could pull out of the soil itself, and yeah, we didn't really try and describe the paleovegetation with no real prior knowledge of what exactly might have been there. But with that said, I think Rachel has some very interesting, well, I don't want to give away the farm here, but I think Rachel has some ideas where we're actually going to begin pulling out plant DNA. In fact, we're already beginning to do that. So now we're pulling out plant DNA in order to try and recreate that paleovegetation, and then determine how much influence that may have, in addition to all these other things that we've measured. So yeah, I mean, that's definitely a good point, and I think we have some tricks to try and get at that in a pretty cool way.
Yeah, if I can pop in,
Sure.
I can say that that's exactly what we're doing. We're extracting the plant reads from the metagenome sequence data and trying to infer paleovegetation, because what's there now is certainly not reflective of what was there, say, in the Pleistocene.
Certainly.
Yeah, so that's exactly what we're doing, and it's just one of those things that we don't currently have funding for right now. So it's every time we have a few spare hours, like, this is what we try and build. So
Sure.
it's coming down the pipe.
Okay, great. Well, thanks for that.
I didn't know how much you wanted to say about that, Rachel, so thanks.
Oh, yeah, sure.
Okay, well, if nobody else has a specific question for Mark, I'm going to turn it over to Daryl Yang, our other co-lead, or one of our other co-leads for the Terrestrial group, to introduce the next speaker.
Sorry, it took a second to start my camera. Thank you, Elizabeth. Thank everyone for staying on. I'm going to introduce Natalie Han-Tels. So Natalie Han-Tels is a research scientist at Lawrence Berkeley National Lab, and her research focuses on understanding the impact of climate change on soil and aquatic biogeochemical cycles via studying of microbial community functions and metabolism. And she's currently leading the Permafrost microbiology component for DOE's next generation experiment Arctic project, which is a multi-phase tactile project to improve our predictive understanding of Arctic ecosystems. Today, she will share some of her work on Arctic, focusing on microbial functional distribution and feedback to climate change and thawing permafrost landscapes. So thank you for joining us, Leslie Han.
Hello, everyone, and thank you, Daryl, for the introduction. My pleasure to be here. So today I have the great pleasure of following Mark. As a talk, normally when I need to give a talk like this, I have to explain in detail what we do and how we do it and why we do it. So thank you very much. And I also would like to start with, oh, let's see what's happening. Okay. Another thanks to, first of all, to our project, NG Arctic. We've been running for around over a decade now. It's a multi-institutional DOE-funded project that comes out of Oak Ridge, but has many other national labs and University of Fairbanks, Alaska, as the team members. We've been benefiting greatly from the resources and infrastructure the Inupiaq communities of the North Slope provides for us. We remain with the greatest and deepest gratitude to them for their support, their understanding to our scientific community, and granting access to their homelands. So Mark really talked about this very nicely in detail, but just when we are looking at the functions in the Arctic, we always need to remember that as microbiologists, our knowledge has a lot of gaps. Harder we try, we are still trying to understand how life actually could happen in frozen environments, yet alone when the just shift happens and changes from a frozen to unfrozen state. Distribution of different materials and moisture really creates a lot of differences in the landscape and creates further complexity to our understanding of what life is under those conditions. So I think just the issue comes from in multitudes of landscape features and moisture distributions and structures, at the end, it really influences the microbial composition and activity. We all are, just more interested in what happens to the methane and carbon dioxide and not nitrous oxide at parts, but, just thaw actually impacts the landscape and the microbes in not just biogeochemistry wise, but also like physically. So today I'll talk about the work that we've been doing in the ice wedge polygons, which are really, just beautiful structures that unfortunately thaw generates, and how we started research on that and what we learned and how it actually nicely ties to some of the things that Mark told you about. So, just the whole idea in here is just that how we are going to fill in the certain critical gaps that we have in our understanding. What we, just know that the permafrost contains a very different microbiome than the Arctic topsoils, which are the active layers. And we know that the thaw changes the permafrost microbial communities. But at the end, we don't necessarily have a good understanding that how that change, just works in different environments. So like the work that Mark showed to you is valuable to, just capture some of those things that we didn't, just understand well enough. But, just we still have to do some work on how that knowledge translates into emissions. So what we know, and this is the history of research, of course, that, just mainly methane and carbon dioxide becomes really, and which one of it is going to be the major resource or, emissions to the system in here. And we, just assume that actually methane is going to be an, just bigger input from saturated environments. And today I'll try to challenge that idea a little bit. So we work in Ecological for a better part of a decade now. And, just landscape looks exactly like this, very classical polygonal tundra. And we've been thinking about how we can approach the question of that, where we'll be finding the microbial thresholds that alter the nature of the rate of the greenhouse gas fluxes, especially when the thaw happens. And when you look at it, even just in flat tundra, we have a lot of structure in the landscape. There are tall lakes, there are trained tall lakes and polygons, and these things are very dynamic. They come and go as the years progress. So what we focused on our understanding is mainly on how we can use the landscape as a unit of understanding. And, they're really very prominent features that have high centers or low centers distributing the moisture quite dynamically, as you see in these figures, like you can stand on them or, just you get sucked into them. So how these structures function in their environment could be quite different. So what we approach to this is that these different polygons indicate different states of the permafrost degradation and moisture distribution, and how we can use this to understand the landscape and the microbial functions. So we used a variety of methods to do this. Most of it really relies on sequencing the DNA, trying to reconstruct genomes and functions a little bit differently than what Mark presented. Besides looking at the genes, we also try to, just put back all the microbes that live in there and take a look at their genomes and how within that structure that we can, just capture certain fine scale differences in them. And then we just do a lot of comparison and analysis to, put a context to our findings. So, but most of our thinking was still driven by, just most of the geophysics work that was done, followed by the chemistry that defined this landscape for us. So when we got there for the sampling to decide how we are going to approach it, we actually knew where the distributions of, just different moisture states and landscape structures were. And then we just took those as landscape units and build upon our study based on that. So first we looked at who was living there in the active layer, in the topsoil, in the more on the superficial side or the more in the mineral close to the permafrost side. So in this side, active layer thickness, elevation, and ice structures is very diverse and just changes very heterogeneously. So we follow their progress through the landscape and find out that each polygonal unit could be defined by their own microbiome. And some of them actually had very similar communities in their organic and mineral layers, shown in the high-centered polygons in here. And some of them actually had very dynamic, very different organic and mineral microbial communities, such as in the low-centered polygons. When we look at the carbon dioxide and methane fluxes coming out of it and how the genes were distributed in this landscape, we consistently found that the carbon dioxide was a larger flux, and the genes that could produce carbon dioxide was very well represented, whereas methane production and oxidation genes were a bit more constrained. Especially, there was a very little methane production potential, even in the mineral layers of higher or more flatter centered polygons, whereas our low-centered polygons, where the moisture is the highest, is the bigger source. Another interesting thing that we looked at with the set was the nitrogen cycle. It usually is assumed that the Arctic soils are nitrogen limited, and in a way that they are. We saw a lot of genes that was, just carried across the landscape that would deal with fixing nitrogen, oxidizing ammonia to nitrite, but most of these things were not really being lost to nitrous oxide, but being circled back to ammonia. So they were not really taking the route that we would initially expect. And there was a lot of investment also to get organic nitrogen converted to ammonia, which seems like another good and consistent source of nitrogen for microbial communities in here. When we look at the carbon decomposition potential, there was a lot of different pathways we could find. So with that in mind, we just really looked at like main resources of carbon, and it seemed to immediately to us like fermentation really came up as a very strong presence in each cases. Just there were a higher fermentation potential in comparison to sugar degradation or hydrolysis of polymers very specifically. So, and this was really well represented across any kind of polygon we looked at. And we could also find these, just trends within the genomes that we could reconstruct at that time, even though they were able to do many different polysaccharides, just the different chitin components. Most of these genomes that we could reconstruct, they were really also able to do quite a lot of fermentation that, just yielding variety of products. So in this study, we could show that the polygon types were indicative of microbial community composition and metabolic potential for the greenhouse gas productions. And most interesting thing that came out of it was that regardless of the soil moisture content, all polygon types were home for facultative anaerobes. So with that in mind, we looked into a little bit more to what happens if we go not horizontally, but vertically to the deeper permafrost, and profiled some of these locations in very much in detail. And the trends that Mark was talking about, you could really see very clearly in here that we also saw this interplay between actinobacteria and other populations. This particular one is a very strong actinobacteria represented location. There is a huge trend that follows the pH. You can almost like put the pH graph to the microbial distribution and just, draw them simultaneously. Because this is a coastal location, we have huge sulfate deposition in here that present both an active layer and very much in the depth of the permafrost, but also has iron, but very little nitrate and nitrite. And when we looked at these parameters in a bit more carefully, we could not necessarily put everything together in a very strict way. So there was a lot of diversity and differences in each depth that we hit in this study. So while depth and pH was the main drivers of this study, just rest of the soil chemistry was not really very deeply explaining to us why the microbial communities are, just appearing as they are. So that goes back to the discussion that Mark was having on the Panarctic scale, that, just paleoclimate that we cannot necessarily immediately understand with soil chemistry measurements is really key in here to, like, just understand this data a little better. But with that, we just went back and just asked us the question that, okay, so now we know our potentials and microbial members. So what does that tell us when we look at the methane and carbon dioxide emissions coming from these systems? And, just how we can understand what can be changing when the thaw happens. So we benefited from the, just the teamwork that was done there. So we have a flux tower that measures the carbon dioxide and methane continuously for us during the summer seasons mainly. And our colleagues in Oak Ridge National Lab, they did a variety of very well-controlled anaerobic incubations, asking the question that, okay, if we take a look at our mineral active layers and compare it to our permafrost, when thaw happens, what comes out from them. So what we know is that when we look at our flux tower data, carbon dioxide and methane ratios are very highly variable. So there's a lot of, like, seasonality in there that, just changes this ratio. When active layer thaws and starts to emit methane and carbon dioxide, that ratios are a bit lower than what happens when permafrost thaws and starts to output methane and carbon dioxide. Especially what we see in this incubations over and over again is that, like, our carbon dioxide to methane ratios coming from the permafrost was really high. So we were mostly emitting carbon dioxide from these systems and not really as much methane, despite the fact that these are very moisture rich, deeply anaerobic and somewhat warmer type of permafrost. So we got the material from one of these studies and used genomics to contrast what's happening in here. So what you're seeing in here, anaerobic lab incubations, looking at active layer and permafrost after thaw and comparing what was the state before they are starting to thaw, and thaw happens and then they go through a whole Arctic summer and what changes in them. So what we saw that especially for permafrost, the dominant actinobacteria that shows in a bit just pinkish reddish color in here, just completely just reduces its size and other communities replaces them. So that this was something just unexpected for us. We really thought that the actinobacteria with just general ability to do many different compounds, they would be able to deal with that. And just there was an quite a bit of similarity between the incubations and the pre-thaw conditions in the active layer, but permafrost samples started to look rather different. So what we found out is that the anaerobic conditions was not consistently yielding or supporting methanogenesis. That's despite the fact that there is a strong fermentation. So we are seeing most of the processes just really being fueled to carbon dioxide instead of being utilized by the methanogens that also exist in the permafrost. So we take a look into this a little bit more, but what we found out is that because the thaw was restructuring fermenters and completely suppressing the methanogens, after some point they were getting outcompeted. So, and it's really kind of interesting in here that if you're an actinobacteria that lives in active layer, you're fine throughout the summer thaw. But if you are an actinobacteria that lives in permafrost and experience thaw and whole summer period, there is a huge difference in those populations that Bacteroidetes and Firmicutes were picking just those processes up. So going back to the question that Mark was also challenging us with, but like who wins? At this particular location, there is a strange combination of competition and balance. So definitely fermentation was winning. with iron reduction and sulfate reduction following through. And most of these processes, especially for in the permafrost, was not really supporting methanogenesis, but feeding other bacteria that could different portions of the fermentation, especially the ones that could take carbohydrates to pyruvate and then to the formate, was really appearing as a winning strategy, where in comparison in the active layer, things were staying a bit more balanced between different pathways that the fermentation could take. So with that, things that we learned that, like, just anaerobic is not going to be immediately methanogenic or methane producing. Anaerobic is probably for this site specifically going to be a lot of fermentation, followed by iron reduction and sulfate reduction and some methane. So as I talked about it a little bit, at this location, denitrification is negligible, but this is not the case for many other sites. And we need to do a bit more research to pinpoint what's happening in here. My guess is it's the sulfate that is, just not really allowing that much denitrification to happen, but just we didn't really look into it very much in depth. So with that, I would like to thank my group, thank the NG Arctic members that, just there were many of them, just did very nice studies and generated data and material for us to take a look into. This project is also in part supported by JGI. And with that, thank you very much and happy to answer any questions.
Thank you, Neslihan. That's a very interesting talk. Does anyone have any specific question to Neslihan? Oh, sorry. Jessica, go for it, please.
Yeah, thanks, Neslihan. That was a great talk. Thank you for sharing those results with us and giving such a nice story. I'm curious about, as you are, about, why you're not seeing the methane in those. And I apologize if I missed that was also a field effect, but I was wondering if in your incubations, part of the reason could just be some sort of I don't know, some disturbance effect that as you, just some mixing in the bottles and things that then you had more of the substrates available for sulfate reduction and iron reduction and fermentation and things. And yeah, could you take me back out to the field as far as applicability for that?
Yes. So, just the one of the good point on lab incubation is that they stop drift, right? So that things gets very intentful. So there is a chance that conditions are not really preferred by the methanogens. That's one thing. The interesting thing for me is that especially the ones in the permafrost seems to be more sensitive to it than in the active layer. So it's not, they are treated in the same way. One survives and the other one doesn't. And during that process, fermentation pathways in one community stays diverse, the other one just kind of narrows down to certain groups. That really seems like an effect that, just driving all this. So when it comes to the field, of course, just when there is mixing, it becomes harder to, just actually limit their environment in the way that we can do in a lab incubation. But with that in mind, that is also I think another experiment that it requires doing that. If we actually force the active layer populations to invade, I know you thought about these questions quite a bit yourself. So I think that seems to be the, just the answer that we should look for.
Thanks. That was really interesting. I appreciate the thoughts on that. Yeah.
Okay,
So,
we have a question in the chat from Randy. Randy, do you want to speak up or you want me to read your question?
Hi. Yeah, I was just curious. You mentioned, you alluded to disturbed areas a couple times as a source of thaw induction, but have you looked at microbial communities or changes in post-fire?
Yeah, so you mean fire? Is that what you mean, Randy?
Yeah, post-fire, have you looked at how they...
Yes, there is one location years ago, Mark and I collaborated that we looked at, not this location, in another one close to Fairbanks in Tanana Valley area. that we looked at in depth what happened to the microbial communities. I can put the link to the chat for you to take a look at. And yes, there is quite a lot of change post-fire in depth profile, also in the topsoils, but that is usually in that location especially was driven by drainage. Again, that's still a moisture question that, just fire, thaw, just because of the mountain slopes that, just things get dry really quickly, and then they have to survive in low activity in really drier soils that used to be permafrost. Yeah, I'll put the paper into chat for you to take a look at. Yeah.
Thank you. More questions? If not, I have a question. I think both to Neslihan and Mark probably. I'm not like a microbial person. I'm very excited to say all this like a spatial distribution of microbial community and correlation with the pH, and also their contribution to methane and carbon production. My question is, we know like with climate warming, with increasing permafrost thaw, there are gonna be increasing activities in microbial, communities. Is there so far, or our current understanding, what are they? Is there or are there any limiting factors that are gonna restrict the activity or function of microbial activities? Because my understanding is they're not gonna just keep doing the stuff they're doing or increasing their activity, and then eventually they're gonna be limited by something that doesn't favor their growth or their function. So any of your perspectives will be appreciated.
Yeah, I think, just at least we have some understanding for the short time, right? But the long time periods becomes really difficult to answer. So at least in the short term, just the importance of soil moisture cannot be really ignored. So, just especially in the field, like drainage features is going to, just determine, just how much activity is going to remain in the system once they thaw. So, and beyond that, we just simply don't know yet. So maybe Mark has some thoughts on that.
Yeah, I mean, my whole point was that, yeah, there could be functional attributes or just taxonomic members that are absent from soils that could limit methanogenesis or something like that, perhaps. But again, it could be just a short-term effect and not a long-term effect. And I think those are perfectly cool types of studies to conduct. And then I think what we're sort of more interested in too is not maybe what limits processes, but are there processes occurring in locations where we hadn't really thought things were occurring before? So we're really focused much more on what's happening in permafrost itself, what's happening in wintertime in talics, and as talics develop, how does microbial activity evolve in those talics? So I think there's a lot more that microbes are actually doing that frequently we're just not really aware of, and there's lots of challenges on other processes too, like nitrous oxide production, for example, seems to be very there are hot spots of nitrous oxide production that occur, and why is that? Is there a microbial linkage to that kind of a process? There's a multitude. There's also timing of thaw. So, is the microbial, the spring thaw. So do the dynamics of microbial processes during spring thaw, do they coincide with, with plant uptake and things like that? Those are interesting. They're not exactly limitations, but they are processes that are occurring that are either new to us or their dynamics are changing. I think those are interesting too.
Thank you. That's very interesting to know. So we have one more question in the chat. Meredith or Elizabeth, do we still have time to let Neslihan answer the question, or?
Yes, I believe we have about 6 minutes left.
Okay. Sorry, I don't know.
Is this my Daryl? Are you asked? Are you?
Oh
asked?
yeah.
Okay, I'm Jack McFarland. I work with Mark Waldrop. That was a great talk. Thanks, Neslihan. So we see a lot of variation, denitrification within the landscape around our work area in Apex, south of Fairbanks, and it's always kind of a what is causing this is a good question, because it would seem that where we wouldn't see denitrification, we see it, and otherwise I'm guess what I'm getting at is it a question of substrate availability rather than more competition between microbes. I've been talking a lot with Tamara Harms about sources of nitrate from rocks, and we know that different rocks have different nitrate content. Granitics, for example, very low, sedimentary, potentially very high. And so I'm wondering if you've ever looked at this in relation to the underlying geology as opposed to maybe a microbial competition. We don't generally see a lot of sulfate reduction in the
Yeah, thank you for that. Nitrogen question was a big one when we were like thinking about that circle, right? So one of the reason, at least microbially speaking, is just that there is not really much of a nitrate being produced that is exuded if you look at the genomes. So, and you do not necessarily see the measure it in the pore water as well to a level that, just there's some inputs that coming from snow, of course, just there could be like some remaining coming from elsewhere, but we never really see in this system particularly nitrate becoming really available. So I think, just that's one thing. There's just maybe simply not enough nitrate being produced with biology or, just deposition itself that would feel that we don't understand it's passed all that well for this particular system, so that maybe it never was. So then, just you end up with this community that, just doesn't really is geared towards denitrification. So when we saw this sort of trend actually, and just busy with the publication, one of the reviewers challenged us quite nicely on that, and we had a data set that we didn't really look at very carefully, and we just tried to measure nitrous oxide, collaborator here in Berkeley lab. So, and they just just fished the entire landscape for over the summer multiple times. They didn't really 150 points, I think, in that landscape, if the memory serves correctly. They didn't really get any nitrous oxide measurements, like measurable amount. So there was really nothing happening in that sense. So I think, just availability of nitrate for the site is one thing. Going back to your carbon question, so since we just are thinking fermentation itself is the main just way of processing a lot of different carbon compounds, most of it doesn't necessarily need to go through denitrification. It seems like just they are already in sort of a semi-competitive balance situation with their iron reducers and sulfate reducers, are that there's just simply not enough maybe carbon to do denitrification on top of everything. When you look at the denitrification that we know, it's usually comes from an excess carbon, excess nitrate situation. It's a costly sort of process when you can actually just grow nicely on your fermentation, maybe just as the energetics of it, you don't necessarily need to denitrify. And could be very different for your location. So that's also something that I really am curious about learning a little bit more in the Arctic, that does it really not matter all that much, or is just this is a coastal thing?
Awesome. I don't think we have more questions, and I'm going to turn it over back to Elizabeth to conclude the seminar.
Well, we're right at the top of the hour, so we don't have time for any more questions, but I want to say thank you very much to both Mark and Neslihan for really interesting talks, and I appreciate the discussion after your guys' talks. So we will be meeting in next month, same time and date, third Thursday of the month. I believe we will be continuing to have another webinar series related to permafrost science. So stay tuned. We'll announce the speakers once they're finalized. Meredith, do you have any final comments?
Expect the recording for this meeting to be posted later today. And that's it.
Okay, great. Well, thank you.
Thank you very much for the invitation again. It's been a pleasure.
Thanks. Bye-bye.
Thanks, everyone.
Thank you, everyone.
Thank you. Bye.
Bye-bye.